Dave. Um, Welcome. It is my pleasure to introduce the first speaker of our CCNB seminar this semester, Dr. Rajiv McCoy, who is in the Department of Biology at Hopkins University. And Dr. McCoy is a very interdisciplinary researcher. And what they do is they develop computational statistical tools to study human genetics. And they are interested in thinking about how germline and somatic mutations shape genome function, reproduction, and development. And so I'm excited to hear your talk, and thank you for coming.
Yeah. Hey, thank you so much for having me. And yeah, it's great to be able to meet so many people whose work I've admired for a long time. And I'm excited to share about some of the work my lab's been doing over the past few years. And I apologize for this kind of vague title. But I was trying to convey that I'm really motivated by evolutionary questions and ideas, and we're studying that at two very different timescales. And I'll tell you a project kind of on either side of that work today. So some of the work we do, just be familiar to many of you, is using data mostly from contemporary genomes of living individuals and looking for patterns in those data and trying to see what it can teach us about the historical evolutionary forces that have shaped our genome, with a particular focus on molecular phenotypes like gene expression and splicing, which we think are kind of an intermediate step toward impacts on larger, higher order organismal phenotypes, things like disease risk or other complex traits. But we're also studying evolutionary changes and genetic changes that are happening over much shorter timescales. For example, in the transmission of DNA from one generation to the next, as well as during the first days of embryonic development, where we think some of the strongest forces of natural selection are actually operating recurrently prior to birth in humans, and we're trying to use genetic data from various clinical genetic tests to gain basic insight into those processes. So starting with some of our work on the more evolutionary functional genomics side, I'll motivate that by zooming out and mentioning some classic work by King and Wilson in the 70s, contrasting the protein sequences of humans and chimpanzees and finding that they're almost identical. And their interpretation of that result was that you must have to invoke non-coding regulatory mutations influencing gene expression in order to explain the phenotypic differences between our species. And that conclusion is largely held up over the decades, and it does not only apply across species, but of course it applies within species. For example, genome-wide association studies, most of the hits from genome-wide association studies are falling in the vast non-coding regions of the genome, and presumably exert their impacts through influences on gene regulation. However, another thing that many of you are very familiar with is that in the field of human genetics, we have these persistent ancestry biases that impact our data sets. This is a figure from 2019 looking at genome-wide association study data sets. And at that time, something like about 80% of the participants in those genome-wide association studies were of predominantly European ancestries with limited representation from these other global populations. And that is a problem if we want to understand the evolutionary forces that have shaped our genomes, but also we might be missing associations that involve loci that are frequency differentiated across populations, or especially rare variation that is sort of by definition recent and therefore geographically stratified. This has gotten a little bit better over recent years, but not dramatically better. And this bias really persists in functional genomic data, for example, gene expression data, where even now, like most of those data sets are from predominantly European ancestry individuals. And so that is, in my view, limiting our understanding of how variation in the level of DNA propagates up to these molecular phenotypes across diverse ancestries and ultimately shapes organismal phenotypes. And so to try to address that issue, my lab recently generated a RNA-seq data set from lymphoblastoid cell lines from 731 individuals from the 1000 Genomes Project. These cell lines are commercially available from Coriell. And importantly, there's already existing high coverage whole genome sequence data, publicly available, open access from those same samples. And so we can intersect those genome sequencing data with the gene expression phenotypes that we can derive from the gene expression data to do this mapping between genetic variants and patterns that we pull out of the gene expression data. So 1000 genomes, many of you are familiar with it, but it spans these 26 globally distributed populations. Of course, there's still gaps and biases even within this data set, but it provides relatively better representation from many of these groups that are really underrepresented in functional genomic studies, for example, East and South Asian individuals or admixed American individuals. This work was led by my former PhD student, Dylan Taylor. And so some of the questions that we want to use these data to address are questions that have been addressed in other studies before, but we think we have unique perspective on this given the diversity of the data. And they include these very basic questions like, how does variation in our genome sequences contribute to differences that we can observe in gene expression and splicing? And then a second question that I'll return to is, how consistent are those effects across different human populations? Is there any evidence that the genetic background modulates the genetic effects on expression or splicing? Okay, so starting with this question, one way that we can get at the impacts of genetic variation on gene expression is to perform association mapping. In the case of these molecular phenotypes, we just operationally limit ourselves to a window around the transcription start site. That's to limit the multiple testing burden, essentially. But we're looking for these cis expression quantitative trait loci, so genetic variants that are associated with the amount of expression of a given gene. And so in this example, the G allele of this SNP is associated with higher expression of this theoretical gene. But you can also derive other molecular phenotypes from these data. One of those is you can measure splicing patterns in different ways. For example, measuring the ratios of particular introns in a given intron cluster. And so, for example, in this case, the G allele is associated with greater usage of this particular intron or splicing out of this exon. So we call the former EQTLs and the latter SQTLs. And so we can map across this data set, which we've called MAGE, all the EQTLs and SQTLs across the genome for genes that are above some minimal threshold of expression. And we pull out examples like this. This is a gene SNHG5, where additional copies of the T allele are associated with higher expression of this gene. But at a global level, of the about 20,000 genes that we tested, we can identify at least one significant EQTL for about 3/4 of them, for a total of about 3.5 million gene EQTL associations, about 2 million unique variants. And we think this is largely just a reflection of our sample size and our statistical power. As we grow these samples bigger and bigger, you'll be able to detect smaller and smaller effects and effects involving rarer and rarer variation. And it is almost certainly true that all genes in the genome have some variation that's modulating its expression. But qualitatively similar results for splicing. Of the about 12,000 genes that we tested, alternatively spliced genes, about 65% of them, we can detect at least one SQTL. Okay, people have done EQTL and SQTL mapping before, right? So what is unique about these data and how can it help us understand these processes? One of the unique advantages of working with a more diverse cohort is it addresses the issue of disentangling causal variants from other variants on the same haplotype in linkage disequilibrium. Of course, in association studies, you don't just pull out one variant, you pull out an entire haplotype of alleles that travel together. And it can be difficult to figure out which one's causal and which one's just along for the ride. But more diverse cohorts effectively capture more recombination events in the history of the sample. And that breaks up these patterns of linkage disequilibrium and allows you to better home in on the causal variant. It gives you better resolution for this procedure that is called fine mapping to try to identify, okay, what are the actual potential causal variants? So we applied a fine mapping approach called SUSY, developed by Matthew Stevens' lab. SUSY has a couple of important aspects. One is that it allows for the possibility of multiple causal variants at a given gene, and that we find that that's really crucial here. And here's some evidence of that. So we see that these independent causal signals for a gene are termed credible sets. SUSY is essentially trying to narrow in, identify a set of variants as small as possible that contains the causal variant with some probability. And so some genes can actually have multiple credible sets, meaning more than one independent association was detected that influences the expression of that gene. And so you can see that within our data, sample size of more than 700 individuals, the modal number of credible sets for these EQTL signals is 1. Well, you have this tail where about 4,000 of the genes, we can detect at least 2 or more independent causal signals. So this is evidence of allelic heterogeneity, multiple common variants that are simultaneously segregating in the population and modulating expression independently. Coming back to the question of the fine mapping resolution, for about 4,000 of those credible sets, SUSY can narrow down the signal just to a single candidate variant with high probability. So we think that these 4,000 variants we've identified with some certainty the actual causal variant that's influencing gene expression. And those are quite interesting to follow up. We have some work that I'm not showing you, intersecting those with epigenomic annotations, for example, to try to understand what's the actual kind of mechanism by which these are influencing expression. Not surprisingly, because we're doing cis-EQTL mapping, many of these are falling within the promoter sequence, but there are also more distant fine mapped QTLs that are enriched in enhancer-like epigenomic annotations that are more dynamic across different cell types. Okay. So it's natural also to consider, like, again, it's not the first EQTL mapping or SQTL mapping study. So what new EQTLs or SQTLs did we discover? And some of them, it's pretty obvious why they weren't discovered before. That includes, for example, this EQTL. So one data set we can contrast with is the GTEx study, which is a multi-tissue gene expression and genome sequencing data set from about 1,000 individuals. So this is an EQTL mapped within this gene PHB2 within our study that was not identified in the GTEx study. The orange are the GTEx p-values. The reason it was not identified is because the minor allele, this A allele, if you look at the frequencies across the different continental groups in an outside data set, or I guess you could also look even within this data set, you see that this variant is restricted or geographically stratified and private, essentially, to East Asian populations. And because that population was not well represented in GTEx, those types of variants are simply going to be missed. So that was one specific example, but we can look at this phenomenon on a more global level. And here I'm visualizing it with a visualization method developed by my postdoc, Arjun Badanda, when he was a PhD student in John November's lab. And the way to look at these are each of these different columns is a different continental group from the 1000 Genomes Project. So African, European, South Asian, East Asian, and add mixed American. And then the height of the bars is telling you the proportion of variance that's falling within a different frequency class, joint frequency class. So C is short for common. So this big block are variants that are common in all of the different continental groups. R stands for rare, and you can't see it, but there's a white box called U, which means that that allele is unobserved in that particular group. And so if we just focus on the eQTLs that we identified in our study that were also identified in GTEx, you can see that most of those are common across all continental groups, which is more broadly true of genetic variation within human populations. But if we contrast that with the eQTLs that are identified in our study, but were not identified by GTEx, which actually turns out to be approximately the same number of eQTL signals, you can see that the frequency patterns are really different for that set, and that the new eQTLs that we identify tend to be more geographically stratified. In particular, there's this big class of variants that are common within the African continental group, but were probably not sampled in the out of Africa migration, and so tend to be rare or unobserved in the other continental groups. However, you also see other patterns of frequency differentiation that we can pick up uniquely in our study. Okay, so coming to this question of the consistency of genetic effects on expression and splicing, one thing that has been a topic of study recently within the field is the possibility of epistatic effects, cis by trans epistatic effects, where the effect of a particular variant could be modulated by the genetic background. And so this is not data. This is just hypothetical example of something like this. But you could imagine that there could be genetic variants where eQTLs, where the slope of the association depends, for example, on the continental group, or essentially picking up frequency differentiated variation in trans that somehow modulates the cis effect. Yeah. Or environment, yes. But in this case, well, we can't totally rule that out. These are lymphoblastoid cell lines that are isolated and grown up in a common environment. So we've tried to mitigate that to the extent that it's possible. Okay, so we wanted to revisit this given the ancestry composition of the sample. And what we find is that when you're evaluating this, it's really crucial to allow for the possibility of multiple causal variants at a given gene. If you don't allow for multiple causal variants, and if you have poor resolution for fine mapping, if you don't narrow in on kind of individual causal variants, you can be easily misled into detecting those cis by trans epistatic effects, or these interaction effects with population. When you allow for multiple causal variants and use the fine mapped QTLs, if we just contrast the slope, so the slopes that I showed on the previous slide, between any pair of populations. So for example, African and South Asian, you see that the genetic effects on expression are remarkably consistent across different genetic backgrounds. And so we think that at least some previous conclusions in the field of these potential epistatic effects or effects of genetic background might have been a sort of technical artifact of lower resolution and not allowing for the possibility of multiple additive causal variants at a given locus. That's not to say that there is no epistasis in the genome. And we do detect some examples that are kind of falling off the diagonal that might be interesting to follow up. But my broad conclusion is that these genetic effects on expression tend to be quite consistent, and additive models do pretty well at modeling the effects. One thing that I didn't show you, but I'll just say, is that there's not much evidence, and people have shown this in other studies as well, there's not much evidence of differential expression of genes across these different populations. The amount of variance in gene expression that's explained by, say, continental group is just a couple percent. And the amount of variance explained by those 26 populations is just a couple percent more than that. And so we're all remarkably consistent, both at the levels of our genomes and our transcriptomes. But there are interesting exceptions to that. Here's one of those exceptions in this gene GSTP1, where this gene is differentially expressed in the individuals from the Peruvian population, lower expressed relative to individuals from other populations in the 1,000 Genomes Project. We can try to interpret this using our eQTL data. So if we pull out the strongest eQTL of GSTP1, we see that it's this C to T polymorphism where additional copies of T are associated with lower expression of this gene. And then finally, if we look at the distribution of that T allele across different human populations, what we see is that that T allele is very frequency differentiated. Many of you work on this, but if you don't, this is a very unusual pattern of frequency differentiation for humans, where you see really large allele frequency differences, and this allele is much higher frequency in the Peruvian population and also this Mexican population relative to other populations around the world, allowing us to mechanistically, or at least make a hypothesis about frequency differentiation of eQTLs driving this differential expression pattern. This is from Los Angeles, I think. Yeah. I didn't make this pie chart, so that's why it's not on. But so that was one example, but we think this also generalizes. So one way of looking at this is to just measure the amount of differential expression between a given focal population, say the African population or continental group, and all the other continental groups within our data set. And so as you move to the right here, these genes are more differentially expressed relative to the other continental groups. And then we can look at the eQTLs, the fine mapped eQTLs of those corresponding genes. And so what we see is that for genes that exhibit more differential expression, their eQTLs are more frequency differentiated between that continental group and the other continental groups, explaining kind of the mechanism of differential expression. Okay, so in conclusion to this first part, variation in gene expression and splicing is greater within versus between human populations. We also see that at the level of allele frequency differences. The exceptions are largely explained by frequency differences in expression and splice altering variants across populations, as opposed to differences in their effects. And the data from these more diverse populations are allowing us to discover new novel eQTLs and SQTLs that were missed before, while improving the mapping resolution to isolate causal variants from the haplotype background. So a couple of directions that we're trying to take this in the future is basically trying to use some of these principles to address questions about the portability of genetic predictions. In addition to detecting these associations, you can then use that information in reverse to try to predict gene expression as a phenotype. And one classic result from the field now is that, genetic predictions that are built on a, one population training sample, for example, Europeans, don't necessarily generalize well to other groups. And through research in the field, we have a good understanding that this is probably mostly driven by differences in linkage disequilibrium. But now that we, can measure these LD differences and potentially use that information to know which genes should have portable predictions and potentially improve the genetic predictions, especially using the higher fine mapping resolution, I think has the potential to improve the genetic predictions and the portability across populations. So that's something we're looking into. In addition, in collaboration with PacBio and Danny Miller's lab at the University of Washington, we're now using it's called Kinnex long read sequencing, RNA sequencing technology to achieve better resolution of isoform diversity from these same cell lines. And so there, rather than getting these short reads that kind of are ambiguously mapping to isoforms, we get a full view, full length view of the isoforms. And we see a lot of isoforms that are not annotated already. And so we're trying to understand that and trying to connect the variation at the level of DNA to these patterns of isoform diversity. And given that we have broad population sampling, interpret that in the context of human demographic history and population structure. Okay, so transitioning now to our work on early embryonic development and prenatal genetics. I'll tell you one recent story there. So one big focus of that work since my PhD has been on the fact that, in human genetics, often a lot of the work, including the work I just told you about, is based on genomes from living individuals. But we also know that in humans, only about half of all of conceptions are thought to survive to birth. And so we want to understand what genetic variation is being lost during that period. And if we can get a broader picture at those earlier stages, what can we learn from that? It's been known for decades that the main cause of pregnancy loss in humans is whole chromosome abnormalities, whole chromosome gains and losses called aneuploidies. And those contribute both to the high rate of clinical miscarriage in humans, but also to this much earlier loss, some of which is, not even clinically recognized, happening even before implantation. So work in both humans and model organisms has established that one major risk factor for aneuploidy involves defects in this process of meiotic crossover recombination, especially in the female germline. And that's because female meiosis, unlike male meiosis, actually starts in fetal development when the homologous chromosomes pair and establish crossovers. But then it arrests in that conformation for decades until puberty and ovulation. So those physical linkages formed by the crossovers, as well as cohesin complexes, which are loaded onto the sister chromatids, are important for stabilizing the paired chromosomes during that decades-long process of meiotic arrest in females. And failure to form these bivalent structures due to abnormal crossovers, as well as age-related deterioration of those cohesin proteins, which are loaded in fetal development but never replenished, are thought to be the main mechanistic causes of these aneuploidies in the female germline. So they can lead to this phenomenon called premature separation of sister chromatids which then causes the sister chromatids to segregate independently in meiosis 1, or this other phenomenon called reverse segregation, where you have segregation of sister chromatids at meiosis 1 and then homologous chromosomes at meiosis 2. So the opposite order that it's supposed to happen. Okay, so with that background, to better understand those processes, my lab has been collaborating with a genetic testing company called Natera to perform a retrospective analysis of single nucleotide polymorphism array-based data from a test called pre-implantation genetic testing for aneuploidy. And the basic idea behind that test is that for in vitro fertilized embryos, they can grow them up to day 5 or day 6 of development, the blastocyst stage of development, when there are these two different cell types, the inner cell mass and the trophectoderm. The trophectoderm will go on to become the placenta, and then biopsy about 5 cells from that trophectoderm layer and do this genotyping, or other companies do sequencing, as well as genotyping of the parents to try to identify chromosome abnormalities in the embryos. And that's used to prioritize chromosomally normal embryos for transfer in IVF and try to improve pregnancy outcomes. Okay, so my lab has been, so whereas those data are collected for individual patients to guide their clinical care, my lab has been aggregating those data into much larger data sets. So here in this particular data set, about 23,000 sets of parents and about 140,000 sets of their embryo biopsies, genotype data from each. Yeah.
This Illumina array, like these sites, like what are they trying to capture?
Yeah, so this is kind of a cytogenetic array where they're trying to kind of evenly space the markers across the chromosomes. It was based on human genome version 18, something like that. So there are big gaps in its coverage. It's also selecting for common variants. So those will be more informative markers for us. But and then I think it has a handful of like kind of disease mutations that are potentially useful in context of another related test called pre-implantation genetic testing for monogenic disorders. Yeah. Okay, so a couple of the questions that we wanted to use these data to address are, again, very basic questions. What patterns of recombination and chromosome mis-segregation are impacting meiosis? And then what's the role of germline genetic variation, inherited variation in the maternal and potentially paternal genome in modulating these processes of recombination and chromosome segregation? Okay. So with the goal of repurposing these data to study these more fundamental genetic mechanisms, my postdoc, Arjun Badanda, developed a Bayesian method to infer both meiotic crossovers and aneuploidies. And so at a high level, that involves using the data from multiple sibling embryos, cases where you have at least 3 sibling embryos, to obtain chromosome level phasing of the parental haplotypes. So to separate out this purple and dark purple and blue and dark blue. And then using a hidden Markov model, we trace paths through the parental haplotypes that best explain the embryo data. And we invoke crossovers as transitions between a given parent's haplotype, right here. Or aneuploidies as additional or missing paths. So here we're taking 3 paths through the maternal, 2 paths through the maternal haplotypes because there's a trisomy of this particular chromosome. So summarizing our results across about 139,000 embryos, we can replicate some really well-established patterns from the field. About 2/3 of these embryos at this stage of development are inferred to be chromosomally normal or euploid. We think that there's actually been quite a lot of selection that's already happened prior to the blastocyst stage. So if you were to look at earlier stages, you'd actually see higher rates of aneuploidy than this. 22% of these embryos have evidence of just a single chromosome missing or extra chromosome. But about 12% have these more, we call them complex aneuploidies or whole genome abnormalities of ploidy. For example, triploidy or what looks like haploidy, but is probably actually genome-wide uniparental isodisomy, two identical copies of the genome. And that happens in a small fraction of cases. Also consistent with the literature, we see that the incidence of aneuploidy varies really substantially across the chromosomes, with chromosome 15, 16, 21, and 22, especially enriched. And we also see that that effect and just aneuploidies in general are really driven by maternal meiotic aneuploidies rather than paternal aneuploidies, which are quite rare. And so again, while it's true that there's been substantial selection prior to the blastocyst stage, we don't actually think that these chromosome-specific enrichments are driven by selection, because those same patterns we see even at earlier stages of development, even in cleavage stage embryos or even in oocytes. So we think that it has something to do with the actual structure of these chromosomes or some other aspect of these chromosomes that's making them susceptible to gain and loss in the maternal germline. The scale of the data also allow us to revisit the relationship between maternal age and potentially paternal age in aneuploidies. And we see that the maternal age relationship is really well fit by a logistic model with a quadratic term for age, which implies that meiotic aneuploidy is accelerating essentially at a constant rate with maternal age. In contrast, paternal meiotic aneuploidies are rare. They affect something like 1% of sperm. People also make similar observations directly from sperm sequencing data. And in this case, we have unique power to be able to study these rare events and test for a paternal age association with paternal meiotic aneuploidy. And we interestingly don't detect any association there, which is a contrast with other forms of mutation that are mostly coming from the paternal germline. Point mutations and structural variation, for example, are enriched for paternal mutations and show a strong paternal age effect. Looking now at the meiotic crossovers, we can also replicate some established patterns from the field, but we get these really kind of clean distributions just due to the scale of the data. The counts of crossovers per embryo are essentially Poisson distributed. And the average number of maternal crossovers that we detect is about 1.5 times the average number of paternal crossovers. Notably, we think that we're missing more of the paternal crossovers because paternal crossovers are enriched toward chromosome ends in the regions of the genome that we have less resolution to detect. Given that we also have parental genotype data, we can perform genome-wide association studies of these various crossover phenotypes that we can derive. For example, the number of crossovers or the proportion of crossovers that are occurring within hotspots of recombination versus outside, to try to identify inherited genetic factors that are modulating the processes of recombination. And so doing that, we detect dozens of associations that were identified in previous studies of living cohorts. So I'm not showing that here, but if you're interested in those, I'm happy to talk about those offline. So one technical challenge for studying crossovers and the relationship with aneuploidy is that some crossovers can be missed when a trisomy involves both reciprocal products of meiosis. So those are effectively invisible to us because we don't have long-read sequencing data or anything that can, capture two heterozygous SNPs simultaneously on either side of the crossover. We're relying on the patterns of haplotype sharing across the siblings to phase the embryos. And so we'll miss crossovers if you have both reciprocal products of meiosis. However, previous work has demonstrated that counts of crossovers are actually positively covariate across chromosomes of the same myocyte, of the same sperm cell or the same egg cell. And we can get into the reasons for that. But using that fact, we can focus our analysis on the disomic chromosomes, on the chromosomes that are normal copy number, and contrast between embryos that either do or do not have an aneuploidy on a different chromosome. And what we see there is that even on the normal disomic chromosomes, aneuploid embryos are depleted of crossovers relative to their euploid counterparts. So that's supporting this, getting around this technical issue of detection of crossovers and supporting a protective effect of crossovers on meiotic aneuploidy risk. Okay. In the last part of the talk, I'm going to focus on trying to identify potential genetic variation in the maternal, in this case, maternal genome that might modulate the fidelity of meiotic chromosome segregation. Okay, and so to get at that, we performed a genome-wide association study of maternal meiotic aneuploidy, where the phenotype was encoded as a binomial phenotype, allowing for overdispersion. The counts of euploid and aneuploid embryos per patient. So effectively modeling the mother's rate of aneuploidy, controlling for the effect of maternal age that I also already described to you before. When we do that, we detect two associations that meet the threshold of genome-wide significance. We've stratified our sample beforehand into sort of a discovery set and a validation set. The reason being that we don't have any comparable data set to be able to validate this in. So this is based on the discovery set. But looking in the replication set, this first association on chromosome 6 does not replicate in that held out set. It also is falling within a large kind of gene desert, and so it's more difficult to interpret. So while we can't rule out that this is something, potentially real that we might want to follow up, I'm going to focus mostly on this second association on chromosome 22. That association replicates in this smaller held out replication set. If we look at the genetic variant involved, again, genome-wide association studies are only powered to detect common variant associations, at least at the scale that we're looking. And accordingly, we see that this variant is common across populations. This C allele is a little bit higher frequency in the African populations relative to non-African populations, potentially reflecting genetic drift associated with the out of Africa bottleneck. We also see, again, consistent with the fact that this is a common variant, we see that the effect size is relatively modest. It's an additive effect that we detect. Each copy of the risk allele is associated with about 1.6% extra risk of aneuploidy for that patient. We detect a small age interaction effect. So the effect gets nominally stronger with age, but that's a very, very small effect. So natural question is what genes could be driving this association? And so using, looking closer at this region, including the imputed markers, we see that the haplotype spans about, 4 genes. But the lead variant falls within this gene SMC1B. And this really is our, very clear candidate, because this gene is extremely exciting to us because that gene encodes a structural component of the meiotic cohesin ring, which I described to you in the introduction, is important for recombination as well as maintaining chromosome pairing during female meiotic arrest. And so classic work by the lab of Pat Hunt and others have shown decades ago that knocking out that gene in mice, for example, causes infertility due to recombination defects, loss of chromosome cohesion, right? That's the cohesin, loss of chromosome cohesion, and ultimately meiotic chromosome mis-segregation. So we wanted to understand what could be the mechanism driving our association. And so we noted that In this case, it didn't look like, well, we didn't find any missense variants in strong LD on that haplotype, suggesting that maybe this could be a non-coding regulatory mechanism driving the association. We also saw that the associated lead variant, or any variant on that haplotype, is an eQTL of the gene SMC1B in diverse tissues from GTEx. That itself is like a little bit weird because I told you this is a meiotic cohesin gene, but it is actually expressed also in these diverse tissues at a very low level in GTEx. But as I already talked about in the first half of the talk, one limitation of studies like GTEx are that they span a narrow scope of human diversity, which limits detection of associations, but also resolution for fine mapping. And so to address that, we turned to those data from the first half of the talk and pulled that gene out of our gene expression data set and see that it's also an eQTL, the same allele, or sorry, the same variant is also an eQTL of SMC1B in lymphoblastoid cell lines from our project. And that opens up the possibility of fine mapping. So we apply this fine mapping approach. We narrow in on 2 independent causal signals at this locus that interestingly are not detectable if you just look at a European ancestry cohort like GTEx, for example. These are in strong LD in European populations, but within the more diverse cohort, you can disentangle these 2 causal signals. One of the causal signals includes 9 candidate SNPs that are kind of distributed throughout this downstream gene with RIBC2. But the other credible set is fine mapped just down to a single variant, and that falls right in the promoter of SMC1B within this open chromatin region that we can see in existing epigenomic data from ovaries. And so we wanted to follow that up. We also see that it's falling within that promoter within a binding motif for a transcription factor called ATF1, which is a activating transcription factor that's expressed in the same ovary data that we drew these data from. So to try to test molecularly for the impact of this SNP on the binding of this ATF1 transcription factor, we collaborated with a lab, Carl Wu's lab in my department, to perform what's called an electrophoretic mobility shift assay, which is a way of testing whether that genetic variant, which is here, it's a substitution from a C allele, which you can see as part of this motif, to an A allele, which is kind of unpreferred within this binding motif, to test whether that has an effect on binding of this transcription factor in vitro. And what we see is that consistent with the eQTL effect, a DNA construct containing the alternative allele, the risk allele for aneuploidy, is associated with lower binding of ATF1 in vitro, suggesting a potential kind of mechanism driving this genetic effect on expression. Okay. Additionally, we're interested in further contextualizing our association, connecting our association results between our aneuploidy GWAS and our recombination GWAS, as well as broadening to other fertility-related traits in humans. And so we grabbed data both from our own study, but also other studies in the literature, extracting the variants that we identified as associated with either aneuploidy or with recombination, and assessed whether they're associated with any of these other phenotypes. And so darker purple indicates more significant negative association. Darker orange indicates more significant positive association. And we polarized all of these according to the aneuploidy increasing allele. And so what we see when we do this is we can identify additional associations. Oh, I shouldn't touch that. Okay. Okay. We can identify additional associations for our recombination GWAS, where these genetic variants are genome-wide significant associated with recombination phenotypes. And while they didn't meet the multiple testing genome-wide significance threshold, we can also pick up smaller associations that we think are probably real with aneuploidy in the same direction. The direction of effect is consistent. Variants that are associated with more crossovers are protective against aneuploidy or vice versa. And some of these same genes, I'm not showing these results, we also pick them up through a complementary approach called transcriptome-wide association study. So these include, I already talked about this meiotic cohesin gene, SMC1B, but we're also pulling out variants within the synaptonemal complex, this temporary zipper-like structure that forms in early meiosis between the homologous chromosomes to mediate the chromosome pairing, as well as these other genes. These are ubiquitin ligase proteins that are involved in the regulation of crossovers. And interestingly, these genes, C14orf39, CCNB1IP1, and RF212, all form kind of an interacting module. Those proteins interact with one another in early meiosis. And so it's interesting that we pull all of those out together. Another interesting observation is that the same variant that's associated with higher rates of aneuploidy and lower rates of crossovers is also associated with seemingly later age at menarche and earlier age at menopause, so shortening of the female reproductive window. And we kind of sporadically pull out other associations like that, but the direction of associations are not necessarily consistent. So we're still trying to understand more mechanistically what could be going on there. The final thing I want to point out is when we look at these other kind of infertility phenotypes, clinical infertility, we don't see any association between our aneuploidy-associated variant and these clinically defined infertility phenotypes, which I think is interesting of itself and is consistent with some other observations from the field and even from my own research, that whereas although it's true that aneuploidy is the main cause of pregnancy loss, I don't think aneuploidy is the main cause of non-age-related infertility. Clinical infertility is very heterogeneous, and aneuploidy is just one, maybe small contributor to it. Yeah, yeah. I haven't specifically. I think, we could do some type of kind of more candidate gene approach to try to lower our multiple testing threshold. We got at that here by just pulling out any variant, any gene and any variant and any gene that's genome-wide significant with some phenotype. But another way to do it is like you're describing, like just take all the meiosis genes and lower our multiple testing threshold that way. I'm wary of that because there's a history in this field of like candidate gene studies that have not replicated well over the decades, but now we're landing on all meiosis genes. So I think it could be valid. Yeah. Of course, we're also interested in trying to understand the evolutionary history of this variant and just this broader puzzle of how you can have genetic variants that are associated with this phenotype that seems so deleterious, right? It's related to survival and reproductive success. How can it become common in the population? But when we look at that, we've tried to look at it in all different kinds of ways. Here, we're looking at using the ancestral recombination graph to try to infer the frequency trajectory of this allele back in time. What we see is that this variant has been common for a long time and it's fluctuated in frequency, but we don't pick up any obvious kind of evidence of either balancing selection or positive selection. The age of the allele is also not particularly remarkable. We time it to about 1 million years old. We see that it's polymorphic within the Neanderthal genomes, for example. So we're not totally sure what's going on there, but we also, my postdoc Arjun developed a second theoretical model focused on a model of negative selection. His idea was that if you assume that, you can model the selection coefficient based on the expected number of viable offspring that a woman could produce over her reproductive lifespan, you would end up with a selection coefficient of about 1%, which is like off the charts. It should not, given the human affected population size, a variant like that should not exist. We shouldn't be seeing it at this frequency. And so that, I think, is just a more mathematical way of saying that that model is not correct. that in humans, we produce few offspring on average, and these pregnancy losses are happening very early in development. There are potentially a lot of opportunities to become pregnant over the reproductive lifespan. And so we need more comprehensive models to really be able to interpret these impacts of aneuploidy and how they translate into something like a selection coefficient. Okay, so in conclusion, I showed you common genetic variation in meiotic machinery is associated with variation in the number and location of crossovers, as well as maternal meiotic origin aneuploidy. That includes this haplotype spanning the meiotic cohesin component SMC1B, where we have this evidence of a non-coding cis regulatory mechanism that drives the effect. But more broadly, I think our findings are highlighting the fact that meiosis has to do multiple things. It has this dual role of generating genetic diversity, creating new allelic combinations, but it's also the crossovers that are involved in recombination are important for ensuring the accuracy of chromosome segregation. And so that's imposing some interesting trade-offs and potentially contributing to, the fidelity of meiosis in the first place. So some directions that I'm hoping to take this in the future include, trying to understand why those chromosomes are so susceptible to aneuploidy. And this might be a little bit crazy, but trying to test potential models of intragenomic conflict and meiotic drive, given that we have these essentially pedigree or family structured data. We can try to ask, are the alleles being transmitted equally to the offspring? And there's a particular interest there in centromeres. While we can't study that directly with these data, maybe we can use some tricks in sequencing-based data to understand the possibility of selfish centromeres and whether aneuploidy could essentially be a side effect of some type of meiotic drive phenomenon. Additionally, I think we're at a really exciting time in this field, and we have a new collaboration started with a company called Cooper Surgical that is doing a different type of pre-implantation genetic testing that's based on exome sequencing, as well as an amplification method that overcomes some of the biases of previous amplification methods involving low input DNA. That, in theory, will allow us to see not just aneuploidies, but smaller forms of mutation, even point mutations, de novo point mutations that could be lethal and could be contributing to the fact that even about a third of chromosomally normal embryos that are transferred ultimately don't lead to successful pregnancies. So there's been work studying living populations and looking for variation that's absent from our genomes and trying to use that to infer the landscape of constraint on our genomes. But here we have potentially the other side of the coin. We can look at the diversity that exists in embryos that just doesn't exist in living populations to understand the landscape of lethal variation within the human genome. Okay, so with that, I want to thank especially my lab, the people that did the work that you saw presented today. Those include Arjun Badanda, postdoc in my lab, and Sarah Karyosha, who led the aneuploidy-related work, and then Dylan Taylor, who led the gene expression work, and our funding and collaborators, and the patients who provided data. And thank you so much for listening.